Hello, everyone, and welcome to today's webinar. Thanks for joining us. Today we'll talk about new methods for CRISPR cell line development and screening using the CellRaft AIR System for automated sorting and cloning. And we have a couple speakers that were joining us today. First, we have Dr. William Buchser, who is a professor in the Department of Genetics at Washington University in St. Louis. His lab studies neurodegenerative disease and other heritable idiopathic diseases. He is also the director of FIVE, Functional Imaging for Variant Elucidation at the McDonald Genome Institute. He and Cell Microsystems were awarded a SBIR, which started in January of 2019. And also speaking will be our own Jacquelyn DuVall, who is a product applications scientist at Cell Microsystems. She has been working on developing and optimizing CRISPR workflows using the CellRaft technology. And quickly before we get started, if you have questions on the right-hand side of your screen there in the GoToWebinar tools, there's an option to type in your question. Please do so at any time, and we will do our best to answer those questions at the end of the webinar. And we will be recording this webinar. So if at the end, we will be able to send the links for those who maybe want to share with your colleagues. And without further ado, I'll hand it off to Jacquelyn to get started.
Okay, great. Thanks, James. So I'd like to start by briefly introducing the CellRaft technology and a few different protocols we've been developing in-house to streamline and automate CRISPR workflows for cell line development and single-cell cloning applications. And I'll then pass the presentation over to Dr. Buchser to discuss the exciting work his lab is doing with CRISPR screening and phenotype unmixing. Okay, so as many of you are most likely aware, there are several major bottlenecks that remain in the cell biology portion of the CRISPR workflow. So what we have here is just a schematic of a really simple and straightforward CRISPR workflow where we introduce the Cas9 and guide RNA into the cells. We then collect cells and allow for recovery, followed by sorting and looking for our phenotype of interest. We then isolate single cells and allow them to expand further into clonal colonies. After that, we can go in and validate that the edit is actually present, and finally do our downstream experiments. And so I'd like to introduce the idea here of automated CRISPR, and we want to start thinking about what would constitute this idea. And so we can break it down into four main proponents here. So first would be sorting. So we'd like to be able to characterize cellular phenotypes after our guide RNA introduction. In terms of cloning, we want to be able to isolate single cells as the founders for our clonal colonies. In terms of expansion, as many of you know, culturing single cells is very tedious and cumbersome, so we'd like to be able to accelerate colony formation. And lastly, in regard to efficiency, we want to be able to do this as quickly as possible with the fewest number of cells and a reduced reagent consumption. A few other considerations here would be we'd like to have a method that would be gentle, so eliminating as many trypsin or scraping steps as possible, no limiting dilution, sheath flow, or microfluidics. And lastly, we'd like to have an option that would be flexible, so something that would be useful for both cell lines and pooled screens. And so for the remainder of this talk, I'd like to focus on how the CellRaft technology can be used to streamline and automate these steps in the overall CRISPR workflow. So the CellRaft technology is comprised of two main parts, the first being our CytoSort array, which is our cell culture consumable. This is an elastomeric microwell array made up of releasable polystyrene cell rafts. So as you can see in the image here, there's a cell raft in each microwell, and this polystyrene is doped with magnetic nanoparticles, rendering it responsive to a magnetic wand. We have two different sizes of the cell raft, so either 100 or 200 microns, and the array is fully compatible with adherent cells And by using any number of standard cell culture coatings, it can be rendered compatible with non-inherent cells as well. So to demonstrate the release and transfer functionality of the CellRaft technology, we have a video here. So the first step is we have a release needle that comes up after we've identified our cell of interest. It punctures the elastomeric base of the microwell, releases the CellRaft, and the magnetic wand transfers that CellRaft into a 96-well plate or a PCR strip tube. So our automated version of the CellRaft technology is called the AIR System, which has a fully integrated microscope with release and transfer hardware. We have 3-channel fluorescence as well as bright field imaging. And we have 2 main run modes here, so real-time or cytometric, both of which have sorting capabilities. And we can sort single cells either in fluorescence or in bright field in order to verify clonality. We have time course imaging, which allows us to analyze images over multiple scans and multiple days. We also have an optional integrated stage-top incubator for time course studies that are a little bit longer, or an application where you'd like to leave the CytoSort array on the deck of the instrument for an extended period of time. We can isolate 96 cells in about an hour and a half, and that includes the initial full array scan where we obtain images from the entire CytoSort array. It also includes the time that it takes to set up your imaging parameters and map your plate and isolate the 96 cells. So for each subsequent 96 cells, we can isolate that in about 30 minutes. And right now we can collect into either 96-well plates or PCR strip tubes in as little as 2.5 microliters for the PCR strip tube. So the first workflow that we wanted to evaluate for the CRISPR sort of application was what we're calling the direct-to-array approach. And so our goal here was to monitor and track a phenotype of interest and capture small clonal colonies. And so here we're tracking a GFP and RFP knockout phenotype, and we're also monitoring clonal colony growth for 4 days on our CytoSort array. In terms of the experimental setup, we're using HEC293 cells that express both GFP and RFP. We're nucleoaffecting those cells with Cas9 RNPs that carry anti-GFP and anti-RFP guide RNAs. And then those cells are seeded directly onto the CytoSort array, where they're cultured for 4 days and then isolated into a 96-well plate for further expansion. In this particular case, we used the CytoSort quad array in order to parallelize the workflow, and we'll discuss that a little bit more later on. So this is an example of what our imaging data looks like. So we're able to scan over time and monitor our phenotype. So what we can see here is up top, what we're looking for is our phenotype to not change. So we want to see the RFP and the GFP signal remain intact over the course of the 4 days, versus down here in our double knockout, what we see on the first day is a strong GFP and RFP signal, and we can see that dim out over the course of the 4 days. So doing this time course imaging allows us to ensure clonality by checking that our colonies actually grew from a single cell. Another important characteristic here is that we can isolate CellRaft at any point during the experiment without disturbing any other cells on the array. So if you go on day 4 and you find a handful of CellRafts that have small clonal colonies that you're interested in isolating, you can do that and leave any other cells on the array intact for further growth over subsequent days, and you can isolate those at any time point. Okay, so as I mentioned, we used the CytoSort quad array to expedite this experiment. So we had 4 different conditions, as you can see here, and we used 4 distinct reservoirs, 1 for each condition. We also have a hexaquad array, which has 24 distinct reservoirs, and that will allow for additional degrees of experimental complexity or more diverse guide pools moving forward. Another important thing to point out here is that we are able to evaluate our experiment each day by putting the CytoSort array back on the AIR system and doing full array scans, which takes about 10 minutes, so we can monitor how well our colonies are growing, make sure everything is remaining clonal. We can look at our phenotype of interest, change the media, etc. So to follow up on that first workflow that we evaluated, we looked at 3 additional workflows for post-transfection cloning. The experimental setup here is very similar, where we're using the same HEC293 cells, the same Cas9 RMPs, and we're doing 3 additional workflows here. The first is a standard method where we're letting the cells recover for 48 hours in a 6-well plate. We're then trypsinizing and collecting 1 million of those to sort singles using FACS. The other 2 methods here, what we're calling AIR Day 0 and AIR Day 1, are both also allowed 48 hours to recover in a 6-well plate. We then trypsinize and seed 2 CytoSort arrays, about 1,000 cells per reservoir in a quad array. And then we're giving one array, the cells have about 2 hours to recover before isolation into a 96-well plate, and the other array, we're allowing the cells to recover for 24 hours on the array before isolation into a 96-well plate. So to pull this all together, what we saw was the highest viability by far came from our direct-to-array approach, with about a 3-fold increase in viability of the transfected cells compared to FACS. And this workflow required about 1,000-fold fewer cells and 10-fold less media. Here we're reporting viability in terms of the number of wells in a 96-well plate that have viable colonies. And so by isolating just one 96-well plate, we can expect to get a couple dozen viable clonal colonies compared to FACS, where we might need to sort 3 or 4 96-well plates to get comparable results. So our main advantages here, we have viable colonies in as little as 1 week post-isolation. All of the workflows using the CellRaft technology that we've shown are at least 48 hours shorter and with fewer trypsin steps. We're also testing similar protocols with iPSCs. And again, as I mentioned, using either a quad or a hexaquad array, we're able to parallelize this workflow to test multiple conditions, pooled guides, things like that. So back to this idea of automated CRISPR and evaluating how well we did. In terms of sorting, we wanted to be able to characterize our cellular phenotype, and we've shown that using imaging on the AIR system, we're able to have a detailed phenotypic characterization. In terms of cloning, we wanted to be able to isolate single cells as founders for our colonies, and we've shown that we can get single cells cultured and identified on the AIR system, as well as ensure clonality. And in terms of expansion, we wanted to accelerate our colony formation, and we've shown that growing colonies directly on the CytoSort array works really well and is the best workflow of those that we've tested in terms of viability for downstream analysis. So to bring this all together in an overall comparison of the AIR system versus other methods that we've tested, in regard to time, on the AIR system, we can have clonal colonies generated in about 4 to 5 days and 4 times that many on a quad array, whereas with other methods, we can expect to get only single cells in about 5 to 6 days. In terms of cell titers, with the AIR system, we're using only about 1,000 cells per reservoir in the quad array versus 1 million for some of the other workflows. Reagent consumption on the AIR system, we use about 500 microliters of media per reservoir. So for 4 different cell lines, we could expect to use around 5 mils or so of media versus upwards of 25 mils with some of the other workflows that we tested. And then in terms of efficiency with the AIR system, what we achieve here is workflow integration. So it's a completely hands-free protocol after the cells are seeded if we are allowing them to culture on the array. And with other methods, we can expect multiple instruments and trypsinization steps to be required. So with that, I would like to encourage everyone to please check out our website and Twitter for updates. We are constantly posting new application notes and publications, as well as workshop announcements and technology descriptions. And with that, I would like to pass things off to Dr. Buchser to discuss his work using the CellRaft technology.
Hey everyone, thanks for joining. We're switching over control. Okay, hopefully you can see what I'm showing you now. Thanks so much, Jacquelyn, for talking to everybody about this. We've been really excited to get this system into our lab, and we've been doing single cell, growing up single cells after CRISPR experiments for a number of years now, about 4 years, and doing the, basically using a classic flow cytometer sorter to do it. And this has really changed our pipeline. It's really, really great. Let me give you a little bit of background from where I'm coming from. So I'm interested in functional genomics. And, here at WashU about 20 years ago, there was a lot of effort spent at the MGI and at WashU to try to sequence the human genome. And since then, there's been a lot of more next generation sequencing, and there's just all, people are getting lots of sequences from disease versus control and all over the world. But the tricky thing about that is that the GWASs that come out of this, right, where you say like, oh, this gene or this variant is associated with this disease, is that as bigger and bigger GWASs are being done, it's just getting very complicated. What we're seeing is that there's even a lot of things that we thought were monogenic are now turning out to be polygenic, and just the interestingness and the complexity around how genes influence disease. It's definitely polygenic at this point for many diseases, especially the ones we're interested in, neurodegenerative diseases, cancer. And we're having to find new ways other than association studies to really pin down what's going on in individual people. And so, if we go out and we take patients in this example, I'm going to show you, it happens to be people with neurodegenerative disease, and we sequence their whole genome or the whole exome. What you find is that most of the mutations or variants that they have are just, they're of uncertain significance, right? So no one has been able to say for sure that they're pathogenic or benign. So the majority of the variants that are out there right now in diseases that we're interested in, probably that you're interested in, are like this, where people may have a mutation in a gene that's known to be important, but we don't even know whether that mutation is functional. So what I care about and what functional genomics is really about is trying to figure out, are those variants, are they functional or not? When you change this position in a particular gene or even intergenic region, wherever you want, does it change the cell or the organ or the patient? I'm specifically interested in how cells respond to this stress. And when we thought about trying to figure out a way to scale this up and to look at larger scale, do functional genomics, we knew we were going to have to look for complex and subtle phenotypes. So we didn't want to look at later stages when cells are just dying or when in cancer growth rate is affected. We wanted to look at earlier stages when things are going on that are really interesting, like changes in synapse, changes in neuroid outgrowth, changes in degeneration. But all these things are very hard to look at. And we knew to scale this up, we'd have to do it at the level of a cell. And so we knew it had to be cell-based. So about 2.5 years ago, we started looking for companies to partner with, and our requirements were just those things. We needed a cell-based way to look at very subtle phenotypes and then have the ability to capture single cells and figure out what variants, what mutations they had. And what we came up with is this great company, Microsystems, which we now have an SBIR with, and we've been making some great progress. And so we use the MicroRaft technology in our lab in 3 different ways. So the first is to do single cell cloning. So this is exactly what Jacquelyn was telling you about in terms of trying to go from a pool of edited cells down to a single cell that has a specific genotype, perhaps also a specific phenotype. But then what we'd also like to do is on this larger scale is phenotype unmixing, which is basically a way to do functional genomics. Another cool thing that we can do in the lab is to use these arrays to work with special cell types. For example, primary neurons love to grow on these arrays, and we have amazing images of these axons, and we can perturb just the axon and leave the neuron alone, as well as co-culture experiments where we have cancer cells and stroma all growing together. We can do really cool both phenotype and molecular biology on those cells. But for the first part of this, I'm going to tell you about single cell cloning, following up from what Jacquelyn said. So you want to do genome editing, you want to knock out a gene, you want to get some kind of a functional edit. And here's some of the concerns around that. And again, we've been doing this for about 4 years, and usually using a flow cytometer on the back end. And so we use ribonuclear protein complexes. So the guide RNA is a synthetic guide RNA. It's mixed with a Cas9 protein, and then we transfect that into cells. What you get out of that is a pool of cells. And that pool has several different kinds of cells in there. Some that are, of course, not transfected at all. Some that are transfected or edited, but the protein is still functional. So this could be because of like an in-frame insertion or deletion. And then, of course, there's a variety of cells that are edited. And so they're usually edited because of a small insertion or deletion. And if that's out of frame, that will often cause the protein to be non-functional. But there's lots of different varieties of that. So out of a single transfection of a guide RNA Cas9, you'll get a variety of different genotypes. And this is the reason why you then have to do single cell cloning, because you'd like to be able to just study one particular genotype to keep everything really nice and clean. And so that's our goal. Our goal is out of this pool of a variety of different types of genotypes is to select one, grow that up, know what it is, and be able to work with it. So let me start out telling you what we've done the most, which is using flow cytometry. And we've over the years really tried to optimize this to make it work as well as it can, and it works. But being able to do it on the Raft array has really changed the way we do things. But here's the way we used to do it. So on day one, we transfect. We then sort the cells out onto 4 96-hole plates. So ideally, best case scenario, we'd have about 400 clones to work with. It takes quite a long time for those single cells to grow out into a colony that we can work with. Our best cell lines take about 12 days. Our worst ones can take 3 weeks to grow out. For cell lines that are really hardy, like HEK293, K562, we can get somewhere in this range. About half of the 96-hole plate has colonies that are viable and are big enough that we can work with. Many other cell lines that we've worked with, it's way lower than this. It's in the range of 20% that can grow into colonies. So after these grow, now is the really kind of annoying part, so you have to trypsinize those 4 plates. And you got to do this, with some kind of like a stamper so that you don't get cross-contamination between the wells. You can't pull, right? So you trypsinize these, you spin it down, you resuspend. About 80% of the cells we're going to take to a lysis plate, and about 20% are going to get reseeded onto 4 96-hole plates to keep them growing, right? Because we don't yet know which ones have the genotype we're interested in. We don't even know what the genotypes are at this point. And so the ones that go into lysis buffer go into our genotyping pipeline, which I'll show you in a moment. And the ones that we reseed continue to grow so that we can select those live cells to maintain in the future. So this works. It takes a bit of time. And unfortunately, it takes 4 96-hole plates and then this slightly annoying trypsinization spin down step at the end. So if you think about how to do this on the CytoSort array, and this is kind of what Jacquelyn was showing you, what we see is that you can now, rather than having to put a single cell in this huge well of a 96-hole plate, we can now put it with its friends at relatively high density onto this Raft array, the CytoSort array. And so at day one, you can see you can't have them too dense, right? You don't want the cells to be too close together, but they can be relatively close together. And this makes them much more likely to survive and to start to form a colony. And so, like Jacquelyn said, you can watch this happen over time, and so you can confirm that, okay, that large colony here actually arose from a single cell, whereas this one on the top probably arose from 2 cells, so we won't pick that Okay, so comparing this directly side by side to the sorting approach, which we've done the most. And so on that first day, we go straight onto the CytoSort array. And then we let the colonies grow for about 4 to 6 days. And so here I'm having on day 6, we use the AIR system to go in and automatically pick out those colonies and put them seed them onto the growth plate. So rather than seeding a single cell, we're seeding this whole little micro colony, as it were. And so by the end, by day 12, it still takes about the same amount of time, right? Because it takes a few days for it to grow on the Raft array. And then it takes a few days for those to grow up into the plate. But now the big advantage is we have the majority of the plate has usable colonies. So we easily pick half as many plates when we do it with the AIR system than we use with the flow cytometer. So that's already half as much work. But unfortunately, we still have to go and trypsinize and spin down and basically split these in part or copy them so we can do the genotyping versus to keep them going. Even if this was the only advantage of the AIR system, we still have moved to this because it's just so much more effective. Like she said, we save on the number of cells we start out with, we save in a lot of reagents, and we get a lot more colonies to pick at the end of the day. But it gets even better than this. And this is something that Jacquelyn didn't mention because we just started doing this over the last few months. Before I show you the new thing, this is just an example of what this looks like. So this is zoomed into a 96-fold plate. This is only about 3 or 4 days after taking the raft, which you can see here on the bottom of the plate. We call it a little slice of butter. It's like a butter on top of a pancake. And so this happens to be for some control cells and some edited cells. And those cells grow out and are very quick to form really nice colonies. Okay, but here's the way that we've moved to. So this has really saved a lot of time and a lot of energy. So we call this the double pick pipeline. And so as before, we do the transfection of the guide RNA and the Cas9, put those cells on an array, and over about 4 to 6 days, they grow out. So now is really where the magic comes in. So if we let them grow out beyond a single raft, we can do something that really helps a lot time-wise. So at day 6, we now, as before, we can take one part of the raft and grow it, put it on a growth plate. But we can take another part of that same colony, the adjacent raft, and put that into the genotyping plate. So this is fantastic because it means we don't have to take the growth plates and trypsinize them and copy them, which again, it causes a lot of trouble. So at day 6, we already can start our genotyping pipeline because we've taken a small part of this raft onto our lysis plate. And then meanwhile, while we're doing our genotyping, our growth plate is growing up over the next 6 days, and we can just go in and take only the 1 or 2 clones that we're interested in carrying forward. And so this is fantastic. And I'll just show you an example from yesterday. We were just doing this, and I said, hey, grab me a picture where you can see this. And so here's at day 2, you can see, starting out with small cell numbers, and then over time, this happens to be only by day 4, you get these HEK cells growing out of a single raft onto an adjacent one. And then this one got picked into a lysis buffer. This one got picked onto a plate. Up here is an example of a colony that we wouldn't pick because we can see it came from 2 separate cells. And so again, so we can get to the genotyping stage in just 6 days, and then this is really wonderful. It's really helped a lot of things that we do. Let me tell you a little bit about the genotyping workflow. So once after we do pick into lysis buffer, we actually pick right into a PCR plate that has a little bit of lysis buffer in each well. Then that whole PCR plate goes into a pre-amplification step because we want to be able to do this on just single cells. We can do it on either single cells or many cells per raft. We go through a cleanup step. And then there's basically 2 steps to PCR that prepare the sequences for multiplex. So this is, we're doing DNA amplification here, right? In this case, we don't care at all about the RNA. We're just looking for the genomic edits, right? So this is PCR to genomic DNA, which is only 2 copies of most genes. So we have to, the front part of this pipeline has to be optimized for your lab to make sure, because it's a very low yield input, right? You're not starting with You're not looking for RNA where there's many copies. You're looking at DNA, which is only 2 copies. But so after you do this, you get this multiplex of all of your, usually many plates worth. Everything's got its adapters. You go and sequence the multiplex and then do your library analysis. Here's what this often looks like. So for any particular genomic target where we've targeted our CRISPR-Cas9, here's where the guide RNA would fall. The triangle is where the cut site would be. Oops. So we have primers that have tails on them. You can see the purple and the green are the tails. And so this is trying to make an amplicon that's about 150 to 250 base pairs long. The first step of PCR is where the regular primers come in, and then the second step is where we add the forward and reverse Illumina indexes. And so what we end out is with this amplicon, and after demultiplexed, we can just read this amplicon directly. And the tailed primers and the indices are what lets us build the library. So the library prep basically just happens as part of what we do in the lab. The reason I have this label as control region and guide RNA region is because one very quick way to determine your guide RNA's efficiency, right, the activity of the guide RNA, is to get sequence that matches a region, usually 30 or 40 base pairs upstream of where the guide RNA is supposed to attack. And so that would be a 20-mer. We're just looking for, in this read, do we find that 20-mer to be perfectly intact? And then we can also do the same thing at the guide RNA itself. Is the guide RNA's sequence perfectly intact? And so a ratio of these two, if the cells were completely unedited, we'd expect a 50-50, right? We'd find reads that both the control region and the guide RNA region are 100% perfect, minus some sequencing noise. But then in edited cells, this sequence would no longer appear because it's edited, and we only see the control sequence as an intact sequence. So that gives us a really quick ratio of just saying like, oh, what's our editing efficiency? Again, the great thing, this is NGS, we're doing it from single cells, so we know not only just is it edited at all, but we know the exact genomic consequence of this edit for every single cell colony that we've grown up. And so that's it. So that's our way of doing single cell cloning after CRISPR. It works really well. And if you guys are doing CRISPR work, I hope that you try it. It's very easy. Just plate these cells on the arrays. The instrument picks them off, grows them up in the plates, and it's really helped a lot. It made it a lot easier than the way we used to have to do it. Okay, so let me switch gears a little bit and talk about how this plays into functional genomics. And while I do this, I'll also show you some numbers about how good these various things work. So in functional genomics, we want to basically be able to unmix different phenotypes and figure out what guide RNAs each of those cells have. And so overall, the way this works is you have a pooled guide RNA library. So you have many different guide RNAs, guide RNA 1 through 7 in this case, all in one tube. You transfect the cells, you seed it onto an array. Here I'm using the different colors to indicate different phenotypes, right? So after you do image analysis and classify these different phenotypes, you can do this on the CMS AIR system. It has a microscope built in, or you could do it on any confocal system that you'd like. Give the CMS AIR system the Raft coordinates, capture those cells, go through the genotyping pipeline. Now you know what did we do to them. And so this is a really cool functional genomics pipeline that we're doing. It's tricky, of course. There's a lot of places where there's potential errors that are introduced. So this is kind of what our tolerance stack looks like. So we'd like our cell survival to be pretty high. We'd like our transfection efficiency to be pretty high. The great thing about the Rafts, again, is that since they're, any manipulation you do to the cells in the Raft array, the cells are still sitting on their Raft, right? So you don't have to trypsinize, you don't have to rip them off. They're still basically sitting in their bed, and you just move their whole bed somewhere else. And so in terms of cell survival, this is really great. We worry about things like guide RNAs per cell, which is MOI. Cells per Raft. Cells per Raft is something we can control. Phenotypic penetrance is, of course, very specific to the experiment that either we're doing or you guys are doing. And so if your phenotype, the great thing is we can do very subtle phenotypes, but at some point your phenotype's too subtle and you can't do this experiment. We worry about capture efficiency. The preamplification has some bias. There's some PCR error, and of course there's sequencing error. The great news is that this all works. So here's a quick look at how those errors kind of pile up and create a tolerance stack. So with a 100 by 100 CytoSort array that has 40,000 Rafts, we seed about 45,000 cells. If we use an MOI of 0.4 and then pick 200 cells that we all think are, have the highest phenotype. In this case, this is our phenotypic overlap. So negatives, their phenotype, is relatively mild, and then the positives, they have a stronger phenotype, but there's quite a bit of overlap. In this case, we get a specificity of 90%, and we can get sensitivities even with fairly large libraries, up to about 1,000 guide RNAs that are really good, about 80%. And so even though this is tricky, we've got the errors down to make it feasible. One of those things that you can do is to adjust the cells per raft, or what the cell density is. This already works out great. So when you're doing your single cell cloning experiments, you just use a single pipette and you add the cells to the array. And of course, they're going to distribute themselves in a Poisson distribution, right, just randomly. And so for the single cell pipeline, that works great because you basically add a third of the cells to your ratio of cells to Rafts is about 1 to 3. And it doesn't look like this. It would look much sparser. And that's actually perfect for doing your single cell pipeline. But for doing these experiments, the phenotype and mixing experiments, we'd really like to utilize as many Rafts as we can. And so for that, Poisson is not our friend, right? Because we get a lot of empty Rafts, and then we get a lot of Rafts that have many cells. And so what would be nice is if we could get a more uniform spacing of the cells to the Rafts. We haven't been able to achieve this just yet, and this works fine for us. And I'll show you a little bit more data about that. But what we have found is that if we use multi-channel pipetters, just switching from a single channel to a multi-channel, we eliminate at least the Poisson happening at a larger scale, and we get a little bit more even distribution of the cells. So that's something that you can try if doing this kind of approach where you need tons of cells is important. Again, for the single cell cloning, it's not so important. Here's what Poisson does look like on these arrays. So this is one quad of a CytoSort array. So this array has 6,400 Rafts in it. And so this is, on the left, is the percent of Rafts that have, in this case, one cell. And so you can see that, if you add a very small number of cells, that, there's about a quarter of the Rafts have a single cell. And if you add more, up to about 1 to 1, that's where you get kind of the optimal number of Rafts with a single cell. For single cell cloning, you really don't want Rafts that have 2 cells or 3 cells or 4 cells. So that's why you want to go to the 1 to 3 number, which is down here, and that's perfect. For our experiments where we're doing phenotype and mixing, we don't mind 2 cells per Raft. We want to get as many that have 1 cell. So we're usually in this range where we have about a 1 to 1 cells to Raft ratio. Okay, so let me show you the proof of concept experiment so I can give you some more numbers for how this all works. And in this experiment, we're going to start out with an HEK cell, just like Jacquelyn did, that has genomic GFP and RFP. So it's a stable cell line that has GFP and RFP. I've colored in yellow because it expresses both of those. So we're then going to hit it with a pool of guide RNAs. And the result, among other things, is that we're going to get some cells that are green because they're knocking out the RFP or disrupting the RFP, and some cells that are red because it is disrupting the GFP. And those are all mixed together on the array or in a tube, and then we're going to either use the array or sorter to try to genotype them and ask, how good did we do in mixing them? So that's just a proof of concept experiment, and it's a good way to kind of test out this system and see how it works. So here's what it actually looks like. So if you plate these cells that have been edited onto the CytoSort array, this is 100 by 100 microns, each Raft. You can see the rafts here. I've adjusted the contrast so that you can really see the rafts, but normally you would only see the cells. You wouldn't really see the rafts so well. But you can see many examples of, nice green cells on a single raft or a red cell on a raft, and lots of other things as you scan through this plate. And this plate would have had 40,000 rafts, so there's many, many examples of cells here that we can pick. And so the unmixing part is just done by the software. So we just say, okay, we'd like to have, where are the green cells? And we can say green single cells or just any green cells. Where are the red cells? And so this isn't physical unmixing. This is just the software identifying, oh, there's a single cell and it's red. I'm going to put it in this category. And so here you can see that zoomed in a little bit. And now what we're going to do is take those cells and have the CMS AIR system, that box that Jacquelyn showed you with the cool blue lights. It's going to automatically take the examples of the red cells and put them into a PCR plate that already has lysis buffer. And so we're going to do a plate of red cells and a plate of green cells. And remember that these are all mixed, and the software is unmixing them and then putting them in these two plates. So after that, we're doing our whole genotyping pipeline, right, which is the preamplification, the cleanup, the PCRs, the sequencing, and the demultiplexing. And so what we want to know now is, how did we do these cells that we thought were red? We take through this whole pipeline with all of its potential errors. How accurately can we now genotype? Can we genotype confirm that these cells in fact have their GFP genomic sequence edited, where these do not? And so when we do that, this is actually the results, that we only made 2 mistakes. So only 2 of these cells that we thought were red, we found some reads that were GFP intact. All the other ones, all of the reads showed that the GFP was in fact was perturbed, was disrupted. And on the other hand, the cells that we picked that were green and not red, those it turned out to be perfect in this case, where every single cell we found had the fully intact GFP sequence. It did not have a disrupted GFP sequence. And so this is great. And this is, of course, not our first experiment, but after several experiments in optimizing the system, we now have this whole pipeline optimized. And where we're at with the Raft-based approach, so again, if we pick out the red cells, almost 98% of those we can get have the GFP edit and don't look like GFP wild type. And then when we pick green cells, we see that the GFP is intact in 100% of the cases. We did this side by side with the flow sorter. We get slightly better results picking out the red cells. And this is actually expected because the flow cytometer has photomultiplier tubes, which are very, very sensitive. So if there was even just a little bit of green or a little bit of green, basically, this is better at seeing that. And so it's just an issue of detecting the phenotype. Interestingly, the opposite didn't work so well in the sorter. I'm actually not sure how to explain this, but the point is that in terms of this experiment, which you can do on both the Raft and the sorter, they're extremely comparable. And going through this whole genotyping system and doing the PCR, from a single cell and from a genomic site, this works very nicely. The last thing I'll point out on this slide is that, again, we can do this experiment side by side with a Raft-based approach and a sorter, but with other phenotypes. So we're interested in protein localization. We're interested in neurate outgrowth, neurodegeneration. We're interested in cell-cell interactions. Those are all things we can do on a Raft. We simply can't do those on the cell sorter. And the sorter, best case, you could sort out to 96. Whatever phenotypes you have, you could sort them into maybe 96 wells. But you can really sort lots of different unique phenotypes into as many different plates as you want with the Raft approach. Okay, so that's pretty much it for me. I've told you about how we do single cell cloning post CRISPR editing. We used to do that with flow cytometry, and now we switched to doing this with the CMS AIR system. I've also told you about, just recently, we've discovered that we can do what we call our double pick pipeline, where we pick one Raft for a growth plate and an adjacent Raft for a lysis plate, so that we can do our genotyping right away and not have to trypsinize the growth plate. told you about our interest in functional genomics and how we're using phenotype unmixing to do functional genomics. And I showed you a quick proof of principle set of slides where we basically can unmix green and red, not terribly interesting, but it's something that we can compare side by side with a sorter and to show that, you know, the Raft-based approach is easily comparable to the sorting approach. So these are the people that have done a lot of the work. Jack Bramley is a senior scientist in the lab and is an expert at the AIR system. And Colin and Jason are really good both at the molecular biology and the genetics. And so I'm very happy to take any questions and talk to you more about what we're doing. Thank you very much. In the meantime, we have a beautiful white slide of snow to show you. Hopefully, if James is here, he can turn us on to see if anybody has any questions.
Yes. One question that's here is, have you ever worked with non-adherent cells?
Great, yeah.
Yes.
So
Go ahead.
I can do that first, Willie, and then if you have anything to add. So we have, worked with both adherent and non-adherent cells. And as I mentioned earlier, any number of standard cell culture coatings seem to work really well with non-adherent cells on the array. And you can just sort of treat it as you would any sort of cell culture consumable. We have an application note on our website about that. So I'd encourage anybody to check that out. We have some protocols for a couple coatings, Matrigel, fibronectin, lysine, and a few others.
And another thing people do with non-adherent cells is right before you pick, you actually can just coat the whole array with gelatin, which doesn't hurt the cells at all. And then you pick, and then you've maintained the cells. You don't let them like slide over to adjacent rafts.
Okay, great. There's a question about working with iPSCs. Let's see. Are you getting good results or similar results with iPSCs?
So in our lab,
go ahead.
Yeah, we can grow the iPSCs pretty nicely on the arrays. You know, we have a Matrigel undercoating, and we have some CRISPR edited ones on arrays, but we haven't like really tried to do the whole pipeline where we're doing brand new edits. Our main focus with iPSCs now is just like making sure they're happy and you know, can they express different phenotypes while they're on their arrays, and it looks like they can. But Jacquelyn, I think you guys are working with other people that are doing iPSCs as well.
Yeah, so we've seen a few different labs get really good results with iPSCs and HESCs on our system. We have one lab in particular that was able to get a couple dozen really good CRISPR cell lines in HESCs when some of the other single cell cloning methods that they had tried were failing. So we have systems in a couple stem cell cores, and they're seeing viability rates that are actually really difficult to replicate using any of the other methods that they've tried. So all in all, very promising.
Okay, great. Actually, it looks like that's all the questions we have time for today, but if we didn't get to your question, we'll follow up with you separately and we'll take it from there. So thanks again, everyone, for joining the webinar, and we'll be in touch for the next one. Thank you. Bye-bye.